about a relatively new development, but something that's happening pretty fast in the clinical trials world, and that's the open access movement, open access to clinical trial data, sharing clinical trial data. And as we know, sharing is caring. Oh, come on. Everyone else goes like that and it goes to the wrong slide, I'm touching. Okay, there we go. Hi, Cameron. All right, so we have some objectives for today. We're going to talk about a very high-level committee from the Institute of Medicine that was developed to develop strategies for responsible sharing of clinical trial data. We're going to understand what is happening on the Canadian scene in terms of the tri-agencies and the Tri-Council statements. We're going to look at what's happening right now with sharing of clinical trial data, and I've got one example of published work in this area, which is from the rheumatology literature. This is the report. So this book just came out in, I want to say, just July. I want to say July 2015 from the Institute of Medicine. It's the report on sharing clinical trial data, maximizing benefits, minimizing risk. And I said it was a panel of 2, 4, 6, 8, 10, 12, 13, maybe 13, 14 people. I think the slide has everyone in. And it represents a lot of different stakeholders in this group of 13 people. Here's industries there because here's the Chief Medical Officer for Johnson & Johnson, a large pharmaceutical manufacturer. The President and CEO of the Genetic Alliance, researchers, a professor of law from Duke, president of a hospital, Brigham and Women's in Boston, University of Oxford. From the clinical trials world, David Demetz is the one who writes the textbook we all use around North America when we teach clinical trials, introductory courses. And Stephen Goodman is another luminary in the clinical trials world. Jeffrey Drowson, the editor of New England Journal of Medicine, chief research officer from the U.S. multiple sclerosis people. So it's getting a lot of different types of people together to come up with these recommendations. And to most of us who are researchers, this all sounds really good. I mean, having access to all the data from other people's clinical trials. Sharing is caring, but also could have said there's gold in them dark hills. But because it will accelerate scientific progress, we might be able to look at clinical trial data sets and make additional findings. It'll change how meta-analysis is done because the individual patient data for all the trials will be available. So we'll have a more comprehensive picture of benefits and risks. Ultimately, the whole goal is to improve public health and patient outcomes. And then we might even be able to find new areas of research by analyzing completed clinical trials, developing new hypotheses about mechanisms of disease. That's all the upside. The panel did recognize there were 5 risks or challenges to protect the privacy of the participants and honor their informed consent, to protect the privacy of sponsors, maybe commercial sponsors who might have confidential information with their economic ramifications, to make sure that things are done well in terms of reanalysis of RCTs. We wouldn't want the public to lose confidence in RCTs because other people are getting their hands on the data and making poor analysis and false conclusions. The researchers who perform RCTs themselves need time to analyze their data and recognition for the work that they've done because the data wouldn't be there without them. And research institutions are worried that here comes something else that they might have to pay for. So there's these 5 different challenges, risks, or burdens. All right. They came up with 4 principles in the report to maximize benefits and minimize risk, to respect the participants in clinical trials, to increase the public trust in clinical trials and in sharing data, and to do the sharing in a fair manner. So this is all background, but they came up, and then we'll get to the chase where things are really at right now. They're moving quite quickly. But they came up with 4 recommendations. We won't go through them all in detail, but the first recommendation is this overarching recommendation that stakeholders should foster a culture in which data sharing is the expected norm. Stakeholders should commit to responsible strategies aimed at maximizing the benefits, minimizing the risks, and overcoming the challenges of sharing clinical trial data for all parties. And then this recommendation is broken down by stakeholders. So in terms of changing the culture, they know what they want the funders to do and to support sharing of clinical trial data. They know what they want the disease advocacy groups to do. They know what they want the investigators and sponsors to do. So they want the investigators to build the thinking that their data is going to be shared right into when they design the trials and they manage their trial data. Adopt common data elements so that if we're comparing across clinical trials, there will be certain key core outcome measures that would be standard in an area. Explain to participants during the informed consent process what gets shared and what doesn't get shared with the individual participants, with all the participants, and with regulators, investigators beyond the trial team, as well as discussing the potential risks to privacy and how the risks are going to be mitigated. And as you'll see, investigators should follow recommendation 2, which gives a timeline for when the data should be shared. Research institutions, CHRIM, University of Manitoba, should ensure that the investigators are sharing their data. Should vote to develop mechanisms to do that, consider it in faculty promotion, and provide training for data science, quantitative scientists to facilitate the sharing. The regulators should make one change to the FDA process so that these CSRs don't have individual confidential information as well as information that should be shared. They should harmonize around the world, and they should make sure the IRBs know that the IRBs should be encouraging the sharing of clinical trial data. The IRBs, research ethics committees, or research ethics boards should be providing templates for informed consent to enable responsible data sharing, consider data sharing plans, and adopt protections as they're developed. Journals, they should require that the authors deposit their data sharing plan at the same time they register for trial registration. They're the ones who got trial registration done by saying that they wouldn't publish articles if the trial was not registered before the first patient was enrolled. And now they're asking the journals to use that same mechanism to have data sharing plans submitted in addition to the trial registration form. And journals also should make sure that submitted manuscripts credit the original authors of data, and also should require that secondary analysis by other people also share their data the same way the primary authors are required to share their data. And professional society should also move to support this new culture, require that those submitting abstracts, manuscripts to their society journals will be required that those data, those trials will be shared, and work with their members to develop these common data elements in each field of medicine. So those are sort of the overall, that's the overall cultural recommendation, including what different stakeholders should do. And then recommendation 2, here's the general statement, but it's broken down into specific times to share data from clinical trials. Although you can share earlier, I guess, if you want. But essentially, they're recommending that at the time of trial registration, as I said, the data sharing plan must be deposited before the first patient is enrolled. And then at the time of study completion, and defining study completion as last patient, last observation, LPLO. Like we used to say LPLP, last patient, last visit, but sometimes observations are done without the visits. Maybe you're hooked into a population health database to monitor outcomes for several years beyond visits. So, but at the time of the last observation, when no more data is being collected, summary level results, including adverse events of intervention, should be made publicly available within 12 months of study completion. And lay summaries of the results should be sent to all of the individual study participants at the same time. Now, and the full data package, so the whole protocol, the whole statistical analysis plan, the whole analyzable data set, and the code used in the software to run the statistical analysis, that should be deposited within 18 months of study completion. There's an exception if there's an economic interest from a commercial sponsor. We'll get to that next slide. So publications can occur also and can occur before completion or after completion of the study. We talked about on the last slide what to do at study completion, but maybe study completion is many years off for a long-term follow-up, but there are results that are going to be published from the trial at 6 months or 1 year or something like that. So this are recommending that investigators make available the post-publication data package, which isn't the full data package necessarily, but the analyzable data set and code that supports the findings that went into the manuscript that was submitted, and the tables and figures within 6 months of the publication date. And then, okay, so if it's a regulatory application leading to approval, they say within 30 days after regulatory approval, the post-regulatory data package should be made available. So that includes everything we've talked about above, but also this clinical study report, CSR, after it's redacted to make sure there's no commercial or personal confidential information about one patient in there. You can imagine that FDA has recently approved the drug that Cheryl Greenberg was working on. You can imagine that clinical study report had those individual patients that she saw probably identifiable there because it's such a rare disease. So that shouldn't be shared, but redacting is the way to deal with that. Or 18 months after study completion, whichever occurs later. And then sometimes a company is applying for regulatory approval and they abandoned the project. They don't get the results they were looking for. In that case, the post-regulatory data package should be shared within 18 months, no later than 18 months after final abandonment. And the word final abandonment is there because occasionally a drug may not live up to its promise, and the company may decide to stop development of it, but maybe there's another company that wants to continue and they sell the rights to that drug to the other company. So in that case, they don't have to do anything within the 18-month frame. In fact, they wait until the last company who wants to touch it either gets it approved or abandons it, so final abandonment. Then the question came out about legacy trials, trials that have already been done. And so the benefits would all be the same, as we talked about right at the beginning, but there's some more complexity to sharing old trials. The PIs might lose familiarity with the data set. The key staff may be gone who do the analysis or the data management. And REVs may have trouble, especially if the old consent forms didn't say that the data would be shared, and particularly if the old consent forms reassured them that the data were only going to be analyzed for one purpose and one purpose only, and not sent anywhere beyond the analysis center. So there might be, legacy trials may have some language in the consent form that made a promise. So they suggest that sponsors and PIs decide for old clinical trials on a case-by-case basis whether or not it's able to be implemented, data sharing is able to be implemented. But certainly they would strongly urge the major clinical trials that have changed management to see if they could get their data sets into the sharing world. So that was number 2. and with the timeline. Number 3, holders of clinical trial data should mitigate the risks and enhance the benefits by implementing operating strategies, data use agreements, independent review panels, governance systems, and transparent access. So that's a bit about the data sharing, data use agreements that would have to be put into place. And as the field develops other appropriate techniques beyond data security and de-identification may be developed to help protect privacy further. Independent review panel, lay representatives included. And then just looking ahead to the future, the Institute of Medicine, I guess, the sponsors of the report of that study, are called upon to take the lead to convene an ongoing multi-stakeholder body with global reach and broad representation to really continue to develop this world of clinical trial data sharing on a go-forward basis. This has caught fire very quickly in Canada. So in 2014, I think maybe it was December 2014, so just over a year ago, the Tri-Council policy statement, which governs ethics of human studies in Canada, issued a revision. It was called TCPS 2 2014, and it made some changes to support sharing of clinical trial data. They said in TCPS 2 2014 that researchers are encouraged to make their data available for further analysis or verification by their peers. And this is in the Chapter 11, the Clinical Trials chapter, mindful of protecting privacy and confidentiality. So that's a change encouraged. Still, REB review is required. This is just some other slides when I'm talking to REB people. But they added, no, this doesn't apply here, that's still for when types of research when you don't need REB approval. But they added this clause. Researchers shall seek REB review, but are not required to seek participant consent for research that relies exclusively on the secondary use of non-identifiable information. So if you want to do research by getting your hands on a data set, they're making sure the REBs understand they need to review those studies, but they need to review them from the point of view that consent from the participants of the trial is not required. And they shouldn't be asking investigators to have to think about doing that. The key is non-identifiable information. So the key is really not to have the key. If you're a PI doing a secondary analysis and you have the key, then that's not non-identifiable information because you can put the code yourself. And there's some language about data sharing and suggesting maybe it's better to do what in fact the Manitoba Center for Health Policy does, and instead of sending data, to do the analysis in their organization. Next, so TCPS stands for Tri-Council. On science.gc it talks about tri-agency, and I think they're synonymous, but because they didn't say change TCPS to tri-agency policy statement, but on science.gc.ca, the government website, they talk about a tri-agency statement of principles on digital data management. This is beyond clinical trials now. This is for any study funded by CIHR or NSERC or by SHRC. And they're calling on, it's a draft statement, it came out about 6 months ago, and they're calling on researchers to develop data management plans for preservation, retention, and sharing of data. And brought the handout. Okay. Here you go. Let's go around the room a little bit. Just cover the whole row. There you go. Go for the whole room. There you go. Start this from the back. See how it goes.
So Shayne, Multidiscipline extracurricular clinical trials, either populational studies.
No, this is the tri-agency statement on principles, so it's not even just CIHR, but it's all studies funded by CIHR, CHRIM, if it's passed in this draft plan. Does anyone else need something like that? Sure.
Shayne, I'll take one of yours.
Sure.
Hey, thanks.
So there's expectations about having these data management plans now for all studies. And geneticists have been doing this for a long time in terms of working together in a big consortium and sharing data sets. But beyond clinical trials, it would provide do observational studies as well. Somewhat high level, but again, at the back page, page 3, there's responsibilities and new responsibilities for researchers, for research communities, for research institutions, and research funders. So it says to, more specifically, as to which data should be shared and preserved. Researchers should consider the data needed to validate research findings and results, support replication and reuse, and consider the potential benefit to their own fields of research, fields other than their own, and society at large. That's the wording for deciding if. So they kind of got you. I mean, I guess you could kind of argue if you're deciding that your study's not very important, but then you kind of shoot yourself in the foot in terms of your overall career. So if you're doing important, just saying if you're doing important work, you should be sharing it.
Is there a place yet where you're supposed to post this
That's
data?
a great question. For clinical trials, places are developing. Really question. CHR has been saying for a long time to put bioinformatics data, atomic data, molecular coordinate data into appropriate public databases. But we're starting to get now databases for clinical trials, or repositories for clinical trials. This is borrowed from a presentation I heard a few months back. And then just again for background, just in terms of what gets shared, we start with raw data. And the raw data is on forums in our offices, or our study coordinators' offices, or maybe in computers if we're using paper CRFs or we're using eCRFs. And if there's a data management format called C- that apparently the electronic CRFs are coded in behind the scenes. There can be raw data for clinical trials that are not on the CRFs, like if there's central lab results that are going straight from a lab to the sponsor center without going through the sites, or surveys might go that way as well. There's a standard data format called STDM, I didn't name it. And from that, people then can work with derived data sets, because in your clinical trial, you might have a composite endpoint. So that's not from the standardized raw data set, but you need to create a new variable for that. Or maybe area under the curve, and you need to create a new variable in terms of the pharmacokinetics. So you create these new variables, and then you've got the derived data set. And that's the point where the data set would be de-identified and sent to maybe transformed, and sent to a data sharing repository. So the repositories are out there. I would like to say the US government has one, but it's got a lot of them. NIH itself has made it institute specific. So there's currently 65 repositories at NIH for clinical trial data. And mechanistic data is associated with clinical trial participants. And some of the ones are up there. National Heart, Lung, and Bloods have BioLink. National Institute of Mental Health has one. IMPORT is from the immunology part of NIAID. The Immune Tolerance Network has something called TrialShare. And sort of the difference between a company approach and government approach. So NIH has 65 repositories. Multiple pharmaceutical companies decide to band together just to make one repository for industry. And it's called Clinical Studies Data Request. And last time I looked, they had they had clinical trial individual patient data on 2,100 RCTs. And there's an independent panel that reviews the requests. Data sharing agreements are in place. And so this is new. As of August 31st, 2015, they had 160 requests, and 105 were approved, 11 were rejected, and the rest were in process. The company Johnson & Johnson has their own system, but they contracted out to Yale University, and that's called YODA, the Yale Open Data Access System. Bristol Myers sends their request to a special panel they've contracted from Duke University. Pfizer, I'm not sure what they do, but so people are looking up trials on company websites and writing to companies to ask for this information. Project DataSphere is also very neat. So there's a lot of chemotherapy trials going on now, lots of breakthroughs in cancer in very diverse, multiple mechanistic areas. And while the data are confidential for terms of the regulatory process, they're sharing the control arms early so that you can have multiple trials with multiple data sets of the same question, and you can be looking at differences in the control groups between studies, or are merging together from multiple studies to summarize. So they're hoping to speed up cancer discovery by getting the control data sets out even before the trials are finished. Some data description journals have developed. Nature Group has scientific data. That's a journal that publishes information about the data set and has the data set repository. Some repositories are out there like PLOS. They have the journal articles that are published in PLOS sometimes have data links. You can just click on the link to get the instructions to access the data. And as I've said, if you can't find the trial in a repository, you might still be able to contact the company itself or the repository you know the company uses to find out about getting that data, and partner companies are responding. The data sharing agreements vary. The key thing the investigators need to do in order to get access to the data, is essentially promise they're not going to re-identify the subjects. That's the most important clause in any data sharing agreement. They also have to promise to protect the data, to follow the protocol they've submitted, and to inform the regulators and sponsors if they discover a safety concern that had not been discovered before. So there's a continuum in terms of actual level of access. In some cases, these databases are public with true open access data. You just need to have an email address. And as long as you have an email address, you can register at the site and download the data sets from the clinical trials. Other ones are restricted to the research community and may have approvals required, like approval of the organization of the project. As I mentioned before, the pharmaceutical one clinical study data request requires the research proposal and it coming from an organization that essentially has a valid research ethics board in the States. Actually, that's not required by clinical study data request. That's required by NIMH, the Institute of NIH that deals with mental health data. In-system access only is another way to make it even more secure. There's some repositories that don't let you download any data sets, but give you access to the data sets through their portal. And the portal includes statistical analysis software, R, SAS, other tools as well, so that you can analyze within their system. And you can sometimes even upload other data if you need to merge data together for multiple data sets. You might be able to do that. So one example, NIH, if you want to go to their website, National Institute on Drug Abuse. And if you go to their website, there's something called a tab for data, the data tab, that gives you the list of all the available study data sets that are there. And for each study, it gives a summary page, the PI's name, the release date, the assessment summary, like what different instruments or questionnaires or surveys were used in that RCT. And there's a place you can click on to see all the protocols that use those questionnaires if you wanted to get those results on that specific instrument or questionnaire for multiple clinical trials. And then there's a button you can click on to download the data. A data sharing agreement comes up, then you click OK and pick the format you want the data in. And you also have a button to click on to get the supplementary information, the clinicaltrials.gov record, the study protocol, the data dictionary as to how the variables are understood, blank CRFs, analysis plan, and de-identification information is all there for you to download to do your reanalysis properly. If you just are interested de novo in a certain assessment, you can go to the assessment tab instead of the data tab, and it lists all the assessments, and you can click on the assessment you're interested in, the survey or whatever, and it'll give you all the trials in the repository that use that assessment. It gives some acknowledgement wording for when you're submitting to a journal, acknowledge the original investigators. If you want to jump off now and get some data sets and start writing another paper based on your new analysis, a few precautionary notes. Understand what data you'll need to combine, what if you need collaboration in the platform, what supporting materials are essential. If you need the analysis plan and they don't give it, then you're not going to be able perhaps to replicate the analysis that the original authors did. You should have a written protocol like any other study that you're doing with a realistic timeline. You need to make sure that all of the necessary variables are there. So to calculate GFR, you need to have the age of the patient. But maybe in this study that wasn't done for renal reasons, they just had 10 to 20 years old, 20 to 30 years old, 30 to 40 years old. If that's all the data that's in there about the age of the patient, then you're not going to be able to calculate the GFR. So if that was going to be your outcome measure, it's not feasible to do that study. You have to make sure that the variables are derived satisfactorily. If you're downloading from an immunology data set, the diagnosis of type 1 is probably well made and includes detectable autoantibodies. But if you're doing some other kind of survey that was done that wasn't about diabetes, but maybe just ask people if they were on insulin or not, and call people on insulin type 1 diabetes, then that might not have the quality that you want or the accuracy that you want for using that variable. You need to understand the data structure. There was an example where there were biopsy reports, and you'd think that each biopsy report would have one field with the answer, with the result, but it was some sort of synoptic report where there were multiple fields for each biopsy. Like each biopsy had 15 different fields per patient. And if the people didn't get that, they could have missed important data from the biopsies. You need to make sure that subgroups are clear, ideally in the same number of patients in the data set clinicaltrials.gov has. And you need to understand how they did data de-identification, what they did with outlier measurements into this derived data set. Did they truncate values? Did they eliminate outliers? Did they delete some variables because the subgroups are small and for privacy reasons they weren't allowed to report them? So you need to have some detailed information of the data record before you can jump into this. Also, the data you want may be in multiple repositories. To be extra safe, Mental Health NIMH has separate repositories for the same patient data from the clinical outcomes and from the genetic data. So you'd have to make sure there's a way someone would be willing to link those two data sets together for you if you needed both types of data. And then just the analysis part. First time I've seen a publication from using this approach was in late 2015 from UCSF. And it was a reanalysis of a trial called RAVE. I'm going to look over at Lily because she's the only pediatric rheumatologist in the room. And this was a reanalysis of the rituximab in ANCA-associated vasculitis trial and how they found a marker early treatment. So vasculitis is inflammation of blood vessels. There's a number of diseases that have vasculitis as a major component. And some vasculitises, you have these detectable antibodies to myeloperoxidase or another enzyme, I guess, called proteinase 3, PR3. But the overall group is ANCA, anti-neutrophil cytoplasmic antibodies. And there's two diseases that are quite severe where you have necrotizing vasculitis that have these ANCA antibodies. So two severe necrotizing small vessel ANCA-associated vasculitides. So they call this AAV. But these two rare diseases are granulomatosis with polyangiitis, inflammation of the blood vessels of the heart, and microscopic polyangiitis, so GPA and MPA. And usually people treat these with immunosuppressives, cyclophosphamide and glucocorticoids are the protocol. And back in 2005, a trial was submitted for registration that planned to enroll patients with A, B, ANCA-associated vasculitides, these two diseases. And they were going to compare cyclophosphamide, which blocks DNA replication, versus rituximab, a new biological monoclonal antibody that works against the B cells, essentially. The primary endpoint was complete remission. And for biomarkers, there was flow cytometry done, and the variables in RAVE were analyzed up and down, whatever you analyze things up and down. But all they found was rituximab might be more efficacious with relapsing disease, but there were, it was fairly equivalent. Complete remission in 35% of new patients getting rituximab, or failure to achieve complete remission. So success in 65% of those getting rituximab and 53% of those getting cyclophosphamide. And they couldn't find any predictors, despite all the work they did, as to who would have complete remission and who wouldn't. The first 6 months of data were put into first the ITN trial share repository and later another repository from the immunologists at NIH. So the new investigators, the new team of investigators, hypothesized that the complete responders, those who go into complete remission, remember these are anti-neutrophil antibody-associated diseases, would have changes in their white blood cell compartment that they could detect at the beginning of the course. Flow cytometry was available. They were done at baseline screening and like everything else in clinical trials, month 1, 2, 4, 6, 9, 12, 15, and 18. 15 antibody staining panels per sample. And they had some information about the B cell population that was publicly available in trial share, Immune Tolerance Network. Now these repositories that have flow cytometry data now have some new statistical visualization and analysis tools. Import, for example, has something called FLOC, which stands for Flow Cytometry Clustering Without K. All right, I didn't name it. I don't know what it means. Why without K is so important instead of just calling it FLOC. Okay, so they use this to analyze the cells, their forward scatter, side scatter, and signal intensity, fluorescent signal intensity. Essentially, they're able to measure size and granularity of cells. And I guess what hadn't been looked at before with RAVE, what hadn't been analyzed before, were the granulocytes. So they defined two populations of granulocytes based on how granular they were. High granularity granulocytes and low granularity hypogranular granulocytes. And so they developed something called the granularity index. So I guess if all your granulocytes were low granularity, you'd get -100, and if they were all high granularity, then you'd get +100. And if you had equal numbers, you'd be in between. So what happened with RAVE was, of the 197 participants, 187 had baseline flow cytometry results, mean age 53 years, severe disease at baseline, 137 with a GPA, 48 with MPA, one with indeterminate disease because it's rheumatology, and one missing. And so the primary endpoint was this complete remission. And in RAVE, we said that there was 52% in the cyclophosphamide group and 64% in the rituximab group, and it was a pooled success rate of 58%. They were hoping to find that this granularity index would be a response predictor. I mean, they did a lot of analysis. They re-verified everything that RAVE investigators had done with the 6 month data set. But not only did they replicate everything, they discovered that the granularity index wasn't just a response predictor, it was a predictor of who would respond to which drug. So the people with low granularity indexes at baseline, that was associated with a better response to cyclophosphamide, whereas the high granularity index at baseline was associated with a better response to rituximab. And then there was intermediate level as well. So they identified cutoffs, just hypothesis generating. They didn't have this as a hypothesis going into this. They were just hypothesizing that the granularity index would affect response without looking at differences between the drugs. So now they suggest we can measure granularity index at diagnosis. Physicians dealing with these patients can. And if it's low, then they get a 66% chance of remission if they go with cyclophosphamide. If it's high, they get an 83% with remission with going with rituximab. And if you're in the intermediate group, well, the docs will still have to do what they've been doing before, deciding between cyclophosphamide and rituximab on their own merits beyond success rate. But the success rate would, go from telling people on average you've got a 58% chance of getting a remission to telling them either they've got a 60% or 83% or a 66% remission rate, depending on which group they're in. So this is clinically important for this disease. I think that's it for this slideshow. So we have some time for talking, for questions. But what we went through today was the existence of this movement for open sharing of clinical trial data, the US report, how quickly things are happening in Canada with Tri-Council statements, and where things are at, and one publication that's already happened from it. And I recognize Dr. Simons.
Has there been any precedent about what happens with RUD? Because technically anyone who's involved in collecting data for a project, they're published, it has to go through the RUD. I imagine that's sort of built into the system.
Right. So if you're doing a clinical trial,
and then more on the other side, the people who
or if you want to get access to a clinical trial,
people from somewhere else are going to be
Yes. So
publishing something using data that they'll get RUD approval from their own institution, presumably, but.
Right. So since I sit on the REB, I can tell you that the companies now are putting into the templates a paragraph in their templates for the sites specific to REBs in the consent forms about clinical trial data sharing. So the larger pharmaceutical companies know about this, and the consent language is going into the consent forms telling people that the clinical trial data set is going to be shared beyond the confines of the study sponsored with other academic institutions, other investigator groups, etc.
And already people writing the subsequent paper just sort of accepts that it's gone through REB at a different institution.
Right, yeah. In terms of these data sharing agreements, they would be required between the investigator who's getting their hands on the data set and their own REB, and the repository where the data sharing is kept. So they may have a contractual step to look at getting the data. But at that point, in terms of the primary sites that were involved in the trial, the data is left and gone to the sponsors. So it's not something that the local REBs would control from the primary study. Hi, Dr. Dillitz.
I was rather surprised and pleased to see that in your presentation that pharmaceutical companies are also making the data available as well. But I was kind of curious, like it seems like the US is kind of done a lot to kind of adopt this. But I was wondering, what do you see? Like this still seems to be an implementation stage here in Canada. What are the major barriers, in your opinion, as far as implementation of sharing of trial data here in Canada?
Well, I think it's just a matter of education and information right now, although we're going to have a discussion about this of at the Meister Research Ethics Board group in March, presenting this in March to them. And, but essentially, it's kind of out of the REB hands at the sites that are doing the primary trials. If the if the ICF has the right language, and the investigators at the Central Reporting Center plan on the data repository, there wouldn't be any barrier from the REBs, even if they're not feeling so comfortable with the concepts. It's novelty. It's novel. I mean, it's really just this month where we start to see all the companies submitting to REB have this language in. This month I presented to Cringe Round, so Shelley was there and Monica Woods was there, and really appreciated learning this because it's just coming to them for the first time from multiple sources. But so far, it's with the support of the Tri-Council at the federal level, it seems like it should go fairly smoothly in Canada.
Are you aware of any spillover into basic science? Because I know there's been some journals publishing on how basic science needs to be more planned and designed according to what they do in clinical trials.
Yeah.
And that's happening. Do you see this happening?
Well, it started as basic science if you count genetics as basic science.
Yeah.
But in terms of individual animal research, for example, I don't know. It would be up to the investigators to consider whether or not the animal study has such ramifications that it should be out there for others to replicate and build upon.
But this, you're saying it's any Tri-Council, any answer grant that you do a self-culture models, so you're supposed to
have a data management plan. The data management plan may or may not include sharing, the reason that society wouldn't benefit from the sharing of that data.
But I think, like in a clinical trial, you pretty much have to justify each patient with the disease that was or was not in the trial.
Yes.
We're throwing away so much stuff in basic science that didn't work or we didn't do the experiment right. So we're not like justifying a lot of negative results in basic science. So I don't know how fair that is.
I don't either. This is a draft statement by the tri-agency, so perhaps some people know about it or are giving feedback about that issue.
I was at a microbiome sort of meeting recently, and it was put on by the funder, so it was all the investigators that they fund, and one of the main discussion points was like the funder was saying, okay, for the next round of funding, we're requiring that you tell us how you're going to share your data, because microbiome data specifically. I think people are just really nervous about, well, I spent all this money and resources to generate this microbiome data. Say I publish my first paper, there's still like 10 more papers I can publish, but I've got to put the data out there.
Right.
I think they're worried other people are going to steal and analyze their data, and some of the answers.
Well, they may have legitimate concerns. So the timelines from the Institute of Medicine report were specific for clinical trials, and are set after. And it's only the data that relate to each publication that goes into the repository after the publication is made. The full data set doesn't go until after, I think, what was it, 6, 12, 18 months, last patient, last observation. So
even 6 to 18 months is not a lot of time, depending on what you're
it depends. For clinical trials, it might be if you haven't, figured it out by 18 months, maybe you're not going to.
No, but for stuff like epidemiologics and microbiome, you can be analyzing.
Yeah, so these time limits don't apply for those kinds of studies. I don't know what, I guess at some point there'll be consensus meetings to determine what timing is valid.
I can see from both sides. That you want to protect your data, but on the other hand, you only have so much capacity to analyze.
That's
so
right.
Put it out there for other people, that would be better for science.
For society and science. And at the same time, you want to get your hands on someone else's data too. Maybe. So, speeding things up through sharing. Sharing is caring. Thank you very much.